Topic of the day is biotechnology. By definition, biotechnology is the use of microorganisms to make practical products. So we're looking at making something practical using some sort of microorganism in this. This often uses gene manipulation, which means we are going to move genes around in some method, and by doing that we are able to add a gene or subtract a gene or multiply a gene or something like that inside of an organism. Now, we're going to start out with some very good illustrations of this. The first illustration really goes back to insulin. Insulin, we know, is a protein that is used for the treatment of diabetes. Insulin is a protein that helps cells absorb sugars, and without the proper insulin concentration, cells do not develop sugars, and this creates a problem for cellular growth. When we use it as a treatment, we always have injectable forms, and people who are diabetics often carry around insulin needles. Nowadays, it is very important if you're going to look at controlling this particular disease. Originally, insulin was harvested from pigs. We had to have a source of insulin that was similar to that of a human. Pig insulin is very similar to that of a human. We slaughter pigs for various reasons. And as they did that, they were able to harvest the pancreas, and from the pancreas they were able to get the insulin, and then they were able to process the insulin to use it in human injections. Problem with this is the supply is limited, and that resulted in a high cost of material. So the supply of insulin was not dependent on the need for insulin, it was dependent on the slaughtering of pigs. The other problem you have is the insulin was not human in structure. Therefore, there is some sort of ethical debate on this. If you don't have human insulin, can you inject it into a human? And there are certain people that would cringe at that thought. So what they did was they removed the gene from a human, they inserted it into a plasmid, they put the plasmid into a bacterium, and now the bacterium, 24 hours a day, 7 days a week, 365 days a year, generates insulin, and that insulin can be harvested. That makes the supply not limited, which drives the cost to be very inexpensive. Nowadays, if you can't afford your insulin, there are places that will give it to you, which means that if you decide not to treat insulin-dependent diabetes, it is a choice and not a necessity. One of the next steps they're looking at is taking the gene and replacing it back into pancreatic cells from the original person, and by doing that you could reimplant the pancreatic cells, and that would allow you to cure, so to speak, diabetes. Let's look at another example. This is of a natural pesticide. There is a bacterium called Bacillus thuringiensis that produces a protein that is toxic to insects when it is eaten. Bacillus thuringiensis occurs naturally in the soil. Pesticides, we know, are applied to plants to kill off insects. And when we do that, we know that pesticides can be damaging to the environment. We also know that they are toxic to people. Therefore, anything we can do to limit use would be good. So what they did was they removed the gene from the bacterium, they inserted it into a plasmid, and then placed that plasmid into a plant. And as they did that, what happened is the plant from then on would produce that particular material, which would be toxic to insects if the insects ate it. So the plant now produces the natural insecticide and kills insects that eat the plant. Sounds like a good idea. And where they started out is they started out working on the boll weevil and cotton. If we look at the boll weevil, that's what it looks like. It's a cute little snout beetle. The whole problem is it attacks the cotton boll, which is the place where the cotton comes out, and it lays its eggs on the inside of it. It does a little damage in order to be able to place the egg on the inside. Then the larvae gets in and it goes to town on this, and it consumes the material on the inside, so in effect you get no cotton. When you look at this particular part, you find that it's almost impossible to get a pesticide in there to kill off that insect once it is inside of the boll. We would call this kind of plant a transgenic plant, because you take genes from another organism and you put them into that particular plant. It worked very well on cotton, and as such, the amount of insecticide that is sprayed on cotton was reduced tremendously. The problem is it worked so well on cotton, they tried it on things like corn and other things. I'm not going to go into the ethics of this. if we want to call it that. The benefits from it can be absolutely tremendous. But one thing I do think is important is that you know about this so that you can get your food from a source where you can trust it. One of the things you will find is if an organism is transgenic, or if a food source is transgenic, it must be labeled. And when it is labeled, you should be able to see that on the labeling. So, biotechnology, using microorganisms to create a technological product that is going to be useful to people.